The process of peptide bond formation, the cornerstone of protein synthesis, holds the key to unlocking some of the greatest mysteries of the biological world. So in this video we will discuss what is a peptide bond and how it's formed. To understand the concept of peptide bond formation, let's first discuss about proteins and amino acids. Proteins are composed of numerous subunits called amino acids. An amino acid has 5 parts: acetyl carbon, also known as alpha carbon because it has 4 different groups attached to it. An amino group. A carboxyl group. R group, which represents the specific side chain attached to each amino acid and determines its chemical properties. And lastly, a hydrogen atom.
So now as we know the structure of amino acid, it's easy to understand the formation of a peptide bond. We just need to understand 2 basic concepts. Firstly, peptide bond is a covalent bond formed between carboxyl group of one amino acid and amino group of the other. And secondly, water molecule is released as a result of condensation reaction, during the formation of peptide bond. The condensation reaction is also known as dehydration, as water molecule is eliminated. Finally, a bond between carbonyl carbon and nitrogen, joining 2 amino acids is created, which is known as the peptide bond. Another fact about peptide bonds is that they have partial double bond character, which means they are planar and have very less to no rotation. This property makes peptide bonds more stable and difficult to break. So now as you all know what is a peptide bond and how it's formed, try to answer this question. The answer is 1. Even though the structure you see represents a dipeptide, the peptide bond present in it is only 1. Similarly, in case of tripeptide, 2 peptide bonds are present. Do not confuse numeric prefixes like di or tri of amino acids with the number of peptide bonds. I hope you understood the mechanism of peptide bond formation. Thank you for watching.